DNA is what makes us all different as it codes for our individual traits. For example, people with freckles will have a different genetic sequence than those without, and this is due to having differences in the pigment melanin. In order to obtain the pigment, the DNA within the cell must code for the proteins it needs to create the melanin. This process is called protein synthesis. Protein synthesis is only possible due to two biomolecules, DNA and RNA. So before we jump into how protein synthesis takes place, first we must learn the basic structures of them both. So what is DNA? DNA stands for deoxyribonucleic acid, and this can be split into two parts. Deoxyribose, which is a modified form of the sugar ribose, is used to help create the helix of the DNA strand, and nucleic acid. These are chains of nucleotides which are attached to the sugar phosphate, otherwise known as the helix. Here we have a strand of DNA. The yellow outer spiral is called the double helix, and this is made from a sugar phosphate bond, deoxyribose being the sugar. And in between the helix are the nucleotide rungs, and there are four in total: adenine, thymine, cytosine, and guanine. But have you noticed anything about the rungs? If you look closely, the rungs are split into two parts, and this is very important for protein synthesis. And the colours always match. Green matches with purple, and blue matches with red. And this is because adenine always pairs with thymine, and guanine always pairs with cytosine. Next is RNA, which stands for ribonucleic acid, and similar to DNA, it is a type of nucleic acid and is also made of sugar, only this time the sugar is called ribose. Unlike DNA, however, RNA is a single helix, which is important for protein synthesis, and the rungs are slightly different. Whereas DNA has adenine, thymine, cytosine, and guanine, RNA has adenine, cytosine, and guanine. However, it does not contain thymine, but instead uracil. RNA can also be split into three roles. These are messenger RNA, which carry the genetic message out of the nucleus to the ribosomes, transfer RNA, which transports the amino acids to the ribosomes, and ribosomal RNA, which assembles the amino acids to make polypeptides which help make the protein. So how does the body know whether to give you freckles or not? Well, each cell has a specific sequence of nucleotides that codes for the sequence of amino acids that will make up the protein the cell needs. This is the genetic code. There are 20 amino acids that can be coded in total, and as there are only 4 base pairs and the codes are read as triplets, it means there are 64 base triplets that can be used to code them, meaning each amino acid has numerous code words. Now that we have learned the structures of DNA and RNA, we are now ready to learn the process of protein synthesis. So to start, protein synthesis is split into two stages, the first stage being transcription and the second being translation. Transcription takes place in the nucleus, and here the enzyme DNA helicase is used to unzip the DNA strand by breaking the hydrogen bonds formed between the rungs, causing the DNA strand to split into two pieces. These are called the sense strand and the antisense strand. The sense strand is important as it acts as a template needed to make mRNA. 3 RNA nucleotides pair with their complementary bases, and small hydrogen bonds begin to form between the pairs. As mentioned earlier, RNA does not have thymine, but instead uracil, which is expressed as a U but still pairs with adenine. As the RNA nucleotides start to pair up, the enzyme RNA polymerase follows, forming a sugar phosphate bond for the mRNA strand. Once the RNA polymerase has reached the termination signal, the mRNA strand and polymerase detaches from the template strand, and the two DNA strands are zipped back up and return to their normal structure. The mRNA strand then has to leave the nucleus to get to the ribosomes, but before that can happen, the strand must be spliced. First, small proteins called SNRPs locate the areas in the strand that are not needed. Once located, the SNRPs form with other proteins to form the spliceosomes, which cut out these parts. RNA splicing is used to remove any non-coding portions from the strand called introns, leaving the strand with exons that will be expressed later on. Now the strand is ready to leave the nucleus to the ribosomes. The ribosomes are made up of a mixture of proteins and rRNA, and here the rRNA provides the mRNA with binding sites, which makes the translation process easier. As the mRNA passes through the ribosomes, it reads the base in triplets, otherwise known as the triplet codon, and finds the tRNA that has the complementary base pairs. This is a tRNA molecule. On one end it has an amino acid, and the other the triplet anticodon that pairs with the mRNA codon. The triplet code AUG is used to start translation, and here the tRNA matches with the mRNA. Once it has found the pair, the mRNA continues to pass through the ribosome so the next triplet can be read.
Created using Powtoon.